All right, today we're diving deep into this kind of microscopic battlefield that's happening inside all of us. We're going to look at the incredibly clever and honestly sometimes terrifying ways that bacteria cheat death and survive our best medicines.
You know the drill, right? You get sick, you take your full course of antibiotics, you start to feel better, and you think, whew, it's over. But then, sometimes that infection comes roaring back. So what's going on? Well, the answer is way more complex than you might think. Turns out it's a tale of two totally different survival strategies.
Okay, so on one side, you've got resistance. This is the one we've all heard about, right? The famous supervillain. It's a permanent, genetic change, like a real superpower that gets passed down from parent to child. But then, on the other side, there's its stealthy cousin, persistence. Now, this is different. It's not a permanent change in the DNA. It's more like a temporary survival tactic. Think of it like playing dead until the coast is clear. And the crazy part? Its kids are born totally vulnerable again.
All right, let's start with the usual suspect, the one we think we know pretty well, resistance. This is all about how bacteria get those permanent superpowers we just talked about, turning them from something we can treat into a really major threat. So at its core, acquired resistance is pretty much what it sounds like. It's when a bacterium's DNA literally changes, giving it the ability to defeat the very drugs we designed to kill it. It's a full-on genetic arms race. And you know what? Bacteria are incredibly fast learners.
So how exactly do they learn so fast? Well, there are two main ways. The first is called vertical transfer. That's pretty straightforward. A resistant parent bacterium just passes the gene down to its kids. Simple. But the second way, horizontal transfer, this is where things get wild. Bacteria can literally share, or steal, resistance genes from other bacteria around them.
And check out how they do it. It's not just one method. They have this whole toolkit for sharing genetic secrets. They can physically connect with a little tube. That's called conjugation. They can just slurp up free-floating DNA from their surroundings. That's transformation. They can even get genes delivered to them by a virus, a process called transduction. And get this, there's even a newer one called vesigiduction, where they send out these tiny little bubbles packed with DNA. It's like a biological internet for bacteria. Absolutely amazing.
Okay, so that explains the superbugs. But what about those infections that just keep coming back, even when the bacteria aren't technically resistant? Well, that brings us to our second suspect, the sleeper cells, and their strategy is totally different. It's not about changing their genetics. It's about changing their behavior.
These are called bacterial persisters, and what they do is, a small group of them, just a tiny fraction of the whole population, basically goes into a state of suspended animation. They just stop growing. And since most antibiotics work by attacking growing cells, the drugs just have no effect on them. Then, once the antibiotic course is over and the coast is clear, they wake up and start multiplying again. And this isn't some brand new discovery, by the way. Scientists first noticed this happening all the way back in 1944.
The question is, how does a bacterium even decide to hit the pause button? Well, as you can kind of see from this, it's a whole complex network of internal alarms. The best way to think about it is like an emergency hibernation protocol that gets triggered when the cell is under massive stress, like, from an antibiotic attack.
So let's break down some of those triggers. For one, they have these things called toxin-antitoxin systems. When the cell gets stressed, it can unleash a toxin that just grinds everything to a halt. Then there's the SOS response. If its DNA gets damaged, that can also trigger a shutdown. They also make this special molecule called an alarmone, which is just a great word, right? That basically screams stress throughout the cell and stops all production. And sometimes it's even simpler. The cell's energy levels, its ATP, just drops so low that it's kind of forced into hibernation.
So for decades, the thinking was that these persister cells were completely passive. Just sleeping, waiting for the danger to pass. But here comes the plot twist. What if they aren't sleeping at all? What if some of them are awake and actively fighting back?
And this is where our whole understanding of this has really changed. As this quote from a recent study says, bacteria with active metabolism can maintain persistence. Such bacterial persisters are not passive dormant cells. The key takeaway, the big mind-blowing part, is right there. They are not passive. They are not just sleeping.
So if they're not sleeping, what are they doing? Well, here are two incredible strategies. First, on the left you have what are called efflux pumps. The bacterium literally builds these tiny molecular pumps in its cell membrane to just spit the antibiotic molecules back out as fast as they can get in. But on the right, we've got an even crazier tactic. They just get rid of their disguise. They shed their own cell wall, which is the very thing many antibiotics are designed to attack in the first place. Yeah, this leads us to the wild world of cell wall deficient bacteria, or CWDBs. By basically ditching their outer armor, they make themselves invisible to a whole bunch of our best antibiotics. They become these little soft, blob-like forms that can just ride out the storm. And then when the threat is gone, they just rebuild their walls and get right back to business. It's incredible.
Okay, let's recap. We have bacteria that can develop permanent genetic resistance. And we have bacteria that can just persist by hiding, by sleeping, or even by actively fighting back. So why does all this matter? Well, this is where this cat and mouse game becomes a really serious problem for all of us.
Here's the bottom line. Bacterial persistence is a major reason why antibiotic treatments fail. It's the reason you get those nagging, recurrent infections. And it's a huge part of why biofilms, those slimy, protected little cities that bacteria build, are so ridiculously hard to get rid of.
And listen, this isn't just some theory from a lab. Researchers are finding these cell wall deficient bacteria in patients with real, chronic, and persistent diseases. I mean, just look at this table. They're being linked to everything from recurrent UTIs and chronic bacteriuria all the way to things like sarcoidosis. These things are hiding out inside the body, causing these long-term, really difficult-to-treat problems.
And this brings us to the most dangerous connection of all. Think about it. That small group of persister cells that are hiding out during an antibiotic treatment, they become the perfect incubator. It gives the bacteria a safe space and, more importantly, time to develop those permanent genetic mutations for full-blown resistance. To put it another way, the sleeper cell can literally wake up as a superbug.
So what we're facing here is an enemy that is so much more sophisticated than we used to think. They don't just build up armor, they have these complex strategies to evade us, to hide, to adapt on the fly. And that forces us to ask a really big question as we think about the future of medicine. If this is what they can do, what's our next move?